Hello, everybody. Welcome back to another wonderful biology class. Today, we're going to be looking at molecular genetics as the start of our next unit. So let's go ahead and get into it. This unit's going to have a few different parts to it. Module introduction, which we're doing right now. We're going to have lesson 1 and 2 be combined because we're going to talk about DNA, which is the genetic material of life, right next to the replication of DNA. They both relate pretty closely. And independently, these would both be really small lessons. So I'm going to make them into one kind of big, massive, jumbo lesson. And we'll see how it goes. Then we'll have a lesson on DNA, RNA, and proteins, the central dogma of biology, or at least it used to be. And then we'll finally round it up with gene regulation and mutation. And of course, we'll have some labs at the end just for kicks and giggles. So moving on to that big question for this entire unit. What is DNA and why is it needed for life? You may not know this, but every living organism has DNA within it. So it is considered to be an essential component for something to truly be alive. Now, let's move on to just the DNA, which is the genetic material. And this is going to be our first real focus of study for this particular lesson. And the question for this particular lesson, not just the entire unit, is which experiments led to the discovery of DNA and which led to the structure of DNA, or the discovery of the structure of DNA. So we're going to be looking at experiments here. Get ready for some nerdy stuff. It's going to be great. Our new vocabulary for today is going to be double helix, something I think I've already said previously in one of our earlier modules, but if not, double helix is the term for today. The review vocabulary for today is nucleic acid, which is a macromolecule or a monomer of a macromolecule. And this is a complex biomolecule based on carbon containing a 5-carbon ring, sugar, a phosphate group, and a nitrogenous base. But our definition here says that it's used for the code of life, which is also very true, very good, and we're going to keep that going. All right, so the first scientist we're going to talk about is Mr. Frederick Griffith. And he's famous because he killed mice. Yeah, it's pretty bad. It's not a nice story. Uh, he carried out a series of experiments with bacteria that have a harmful form and a harmless form, and was looking at something called a transformation factor, something that converted the harmless form into the deadly form. And the bacteria he was working on was the pneumoniase bacteria. And he discovered that when he killed some of the bacteria from a dead mouse and put it in with the bacteria that were harmless, it made the bacteria lethal. It turned them into smooth bacteria. So rough became smooth. Rough were easy for the body to grab with a phagocyte, a white blood cell, and consume and kill, whereas the smooth had a slime mold. And the S. pneumoniase bacteria tend to lead to pneumonia, which is fluid in the lungs, and your body's inability to kill the bacteria has to do with the slime coating they produce that make them smooth. Now, after Chase came Oswald Avery, and he identified the molecules that transform the R strain, rough strain, of the S. pneumoniase into the S strain. Okay, so he's the one who figured out what was causing the transformation. Avery concluded that when the S cells were killed, DNA was released. And so the R bacteria incorporated that DNA into their cells and changed into the S strain cells. So they went from rough to smooth and slimy. Nasty, nasty stuff. Okay, after this came Alfred Hershey and Martha Chase. So Hershey and Chase. And they used radioactive labeling to trace bacteriophage DNA and protein, right? So they made some bacteriophages that had the protein that was radioactively traced, meaning that it had, say, carbon 14 or some kind of carbon that had a radioactive component to it that can be seen, and incorporated into the virus. So they tagged the protein and they tagged the DNA. And what they discovered from their experiment is that the phagocytes would inject the DNA into the bacteria, and then the DNA would replicate and the bacteria would die. The radioactively tagged protein from the virus, on the other hand, stayed outside of the bacteria. So it wasn't the protein that was being used to make more viruses. And so this was another really good indicator that the stuff that was communicating, the viral code or the viral information, viral program, was the DNA and not the protein. Now, Erwin Chargaff is a very, very famous fellow. Not only does he have a great name that sounds like a cough, Chargaff, he also analyzed DNA fragments of adenosine, guanine, thymine, and cytosine, A, T, C, and G, and he discovered that they tended to appear in equal amounts. C, cytosine amounts, roughly, not completely, equaled guanine, and thymine, roughly, not 100%, equaled adenine. And so he discovered what's called alternate base pairing. This is also known as Chargaff's rule. Now, Rosalind Franklin and Murick Wilkins did some studies on DNA with a technique called X-ray crystallography, or X-ray diffraction, to discover the structure of the double helix. And this is one of the more sorted stories in the the history of science, because you usually hear that Watson and Crick discovered the three-dimensional structure of DNA, the double helix. Well, it was actually Rosalind Franklin's data that was used to make that discovery. All Watson and Crick did was make a cardboard model and present it. So they borrowed her data to show that DNA has a double helix. It's pretty nasty. She did get a Nobel Prize after she died from cancer. Very sad. But her data was kind of taken from her, which is really sad. And that brings us to James Watson and Crick, who, using Franklin's data without permission and Chargaff's data, I believe that was with permission, they came up together and figured out that DNA had a double helix. And that was the structure. It was like a twisted ladder where phosphates bonded to carbons of a sugar, creating a backbone, and nitrogenous bases alternately paired with each other, forming a ladder structure in the middle. Important features of the model constructed by Watson and Crick include two outside strands of DNA, phosphate to carbon bonding, so the phosphate group to the sugar group, or sugar carbon, of a nucleotide molecule, cytosine and guanine base pairing to one another, and thymine and adenine base pairing to one another in equal amounts. And so you had these very strong phosphate bonds going all the way up and down your DNA vertically, and then horizontally you had hydrogen bonding occurring between your nitrogenous bases, between adenine and thymine, and cytosine and guanine. So again, nucleotides have three basic parts. It has a phosphate group, it has a 5-carbon sugar, and then it either has 1 or 2 nitrogenous rings. Okay, and so your nitrogenous rings determine whether or not it's a purine or a pyrimidine, and whether it's going to be an adenine or a thymine, or a guanine, or a cytosine. And don't stress if this is confusing. We'll look at these a little bit more later. So DNA structure. Well, DNA is a double helix. It has rails going up and down with phosphate bonds along the vertical backbone that are very strong. And then it has those hydrogen bonds across the middle, which individually a hydrogen bond is very, very weak, but it can be unzipped by pulling one bond apart at a time. The DNA molecule's orientation, it has a specific orientation where you have two strands, a 5-to-3 strand, 5-to-3 that goes in one direction of your DNA, and a 3-to-5 strand that goes in the opposite direction. They are said to be what's called anti-parallel, and that's the orientation of DNA. But it's interesting that codes for proteins come from this side and can come from this side as well. So it's not just one side that's important. All right, that's enough for this particular lecture. I've gone on quite a bit. So thank you so much for listening. I've got some questions for you, and it will be good. Thank you.